Hello everyone, my name is Benjamin Samudio. It's May 2023 and I'm going to talk about ReLiEF fingerprints and TMAP for 3D-based molecular similarity visualization. ReLiEF stands for Receptor Ligand Surface Feature Fingerprints for encoding structures and physicochemical properties. So using these fingerprints we can encode interactions, interfaces, voids, and surfaces. Now as an analogy, let's think about the Google search engine. So there we can put a search term into the search bar and we can get results back very quickly. Now Google is able to do this because of a process known as indexing. Now imagine if we had a similar process but for chemical structure. So in this example, red is a protein structure with a ligand bound, and what we want to do is compare it to all other structures. We want a way to gauge its similarity. We want to be able to cluster those structures together, and we want to serve up exemplars as search results. If we had this capability, then for experiments, for example, we could propose new pockets or vectors to pursue. In machine learning, this could help with feature engineering and delineating the domain of applicability, and in simulations this can help guide them into unexplored regions of chemical space. In order to emulate a search engine such as Google, our process would need to be fast to quickly assess similarity, flexible to encode a variety of information, and vast to handle a large amount of data. Now I'm going to talk about a process that I developed to convert protein structures, in this case, to fingerprints, and then take those fingerprints and visualize the similarity of those structures. So converting structures to fingerprints is done by a Python program that I wrote, and that takes about 5 minutes per structure. And then to convert the fingerprints to a visualization of the similarity of those structures, it takes about 40 seconds for everything, and that's done using an open source program called TMAP. Now this makes the process fast. Now the way that my script works is first I align structures to have a common coordinate system. So in this case, able kinase conformations are aligned together. Now for each structure, I render them as a surface. Then I slice a surface into equally spaced segments. And then for each segment, I add intersection points to the segment contours, shown here as points colored by rainbow. Now as an example, I show one segment here and the contour being outlined by green points. And what my script does is it moves the band from the negative y to the positive y side. And as it does that, every intersection with one of those green points is captured into a fingerprint bit as a collection of letters. And those are bins, those represent bins of distance. So for example, A equals 0 to 0.1 angstrom, SF encodes 49.9 to 50 angstrom, and ALL encodes negative 49.9 to negative 50 angstrom. And what's really exciting here is that as long as a property can be encoded in terms of text, then it could be captured into this fingerprint bit. So what this enables is that each bit can encode diverse information simultaneously, such as distances, charges, pharmacophores, functional groups, atom types, etc. So the distance is relative to the z-axis. We can imagine this intersection capturing the distance, but also capturing, for example, what residue is near that point or what pharmacophore is close to that point. That can also be captured at the same time in this fingerprint. So that's what makes this process flexible. Now to give a little bit of background as to how fingerprints are converted into a visualization where we can see the similarity between those fingerprints, that's through a process known as locality-sensitive hashing. So here I show 3 example fingerprints, 1, 2, and 3, and they are composed of single letter bits. So here there are 3 single letter bits per fingerprint, and the gold window is what's known as a shingle-in window. And here the shingle-in window is of size 2, so it's going to move along the fingerprint and capture pairs in this case. So here for the first shingle-in window position, it's going to capture C and G. Then it's going to move 1 over and capture G and D. Now all of these pairs are then collected into what's known as a vocabulary. This is a set. And then that set is going to be used in order to make what's known as one-hot encoded vectors. So here in this case, the pair AD, if it's found in a fingerprint, then that is encoded as a 1, whereas if it is not found in the fingerprint, then it is encoded as a 0. So the one-hot encoded vectors can be very large, but they can also be very sparse. So what we want is a way to represent those fingerprints in a condensed way while still maintaining the differentiability that the one-hot encoded vectors have. So this is where min-hash functions come in. They convert the one-hot encoded vectors into what are known as signatures. Then those signatures are subdivided, and hashing functions are applied to those subdivisions. And the hash buckets, if there's a collision that occurs, then that's what's known as a candidate pair. So in this way, we have the ability to encode structure and physicochemical structures, properties rather. And now we have the ability to be able to determine whether that encoding, those encodings, are similar to each other or not. So now that we have those two capabilities, we're moving on to visualizing the similarity that's calculated with the locality-sensitive hashing. So here I show what's known as a TMAP, and this is a minimum spanning tree, so that points that are close to each other are also very similar. So as an example, I show these two compounds which are very close to each other in their TMAP, and they're different only by one atom. So again, points that are close to each other are going to be very similar. I show a TMAP of 1,000 fingerprints being compared here, but I also show next to it a TMAP of 1 million fingerprints to show that this process can be vast. You can make large comparisons between a large amount of data using this process. So now that we have these capabilities, I want to apply it to something and see how it works. So here I came across 3 distinct Abel conformational states probed by nuclear magnetic resonance. And so there are 3 conformational states that were identified: a ground state, which is the active state, and it has a PDB accession code 6XR6, and then 2 excited states which are inactive, 6XR7, which has a unique structure with an unknown biological function, and a selectively stabilized in the full-length kinase structure 6XRG, which occurs when there's regulation by the regulation module or the fully assembled kinase. So the active structure, the population is about 88%. The inactive and unique structure has a population of about 6, as does the inactive structure, with all the classical hallmarks of a completely inactive kinase. So here I align all of the conformations that belong to those conformational states. So there are 3 conformational states with 20 conformations, each again identified by nuclear magnetic resonance, for a total of 60 conformations. And this is what I'm going to build the TMAP on. So now, having encoded these structures in fingerprints, now I show the TMAP here in the center. So notice that they're colored by the corresponding PDB structure. Again, 6R6 is going to be active, and 6XR7 and 6XRG are both inactive, green being that unique inactive structure. So when I look at the different clusters that are represented in this TMAP, again, recalling that clusters are going to be those structures which are similar to each other. Here in this cluster at the upper left, 6XR7, again, that's the unique inactive structure. And when I align those structures together, they do seem to have very similar structure. Going to the opposite side of the TMAP, here's a cluster of 6XRG, again inactive, but this is again classical, has all the hallmarks rather of an inactive kinase. Looking at that structure, very similar. Those conformations are very similar to each other. But what's also very interesting is when I find these clusters of both active and inactive, so blue and green clusters here together, again, having different designations in terms of their active state. However, when I align those structures together, they are very similar to each other. So this is interesting because even though they have different designations in terms of their active state, they share very similar conformations. So this suggests that maybe TMAP could be used to find those structures which are very similar to each other, even though experimentally they may have different designations. Now here what I do is I align the 6XR6. Again, this is going to be the active structures, the active state conformations rather. I align all of them together and I fill them with the alpha spheres from F pocket. And we're looking down at the end lobe. The allosteric site here is at the bottom. The ATP binding site and hinge region are up here at the upper left. And I color them according to where they are at in the TMAP. So red is at the upper left and violet is at the lower right. And what I find to my eye is that as we go down that TMAP, the alpha spheres they were again found from F pocket seem as if they're receding away from the allosteric site back into the ATP binding site. So there doesn't appear to be a lot of violet here, but there is violet at the ATP binding site, just like there is other colors. Whereas with red, that really seems to encircle the allosteric site and have a presence throughout. Now there also appears to be this differentiation between pocket volumes. So again, working with those same alpha spheres from F pocket, I show the volumes here in magenta. And at the upper left, again correspond to a cluster of 6XR7 structures exclusively. I see that there is volume between the alpha C helix and the beta sheets. Whereas if I go to the opposite end of the TMAP, again showing a cluster of 6XRG, that volume is below the alpha C helix. So there's that difference there, and in between there seem to be different volumes. So there appears to be this clustering of pocket volumes as well. Now zooming in on the cluster that I mentioned, which is again a mix of active and inactive states, I represent those as a heat map to show that in fact they do have the similarity shown in dark blue, whereas there are other regions, like for example the opposite ends of the TMAP, which are less similar, and that's shown here in red. So what are some things that can be done with this? So one, we can encode distances and other information such as pharmacophore. So here I'm showing one of the conformations of Abel kinase that is colored by pharmacophore. Again, because the fingerprints can capture anything that can be encoded in text, we can encode not only the distances within this protein, but also the pharmacophores that exist at certain points within the protein. So all of that can be encoded in the fingerprints. The second thing is that we can explore the intersection of pockets. So at different points in the TMAP, they're going to have different F pocket alpha spheres. Now we can calculate the intersection of those to perhaps find some conserved pockets that exist at different regions or maybe throughout the TMAP. And they may be places where we might want to engineer ligand, for example, because it seems to be conserved across these conformations. The third thing we can do is characterize compound conformations. So for example, we can develop three-dimensional structures for compounds. We can align them together, we can compare their structures together and their physicochemical properties, or we can also explore ligands within binding sites and capture those interactions. Again, as long as it's text-based, it can be incorporated into these fingerprints and we can view the similarity of those. So this is a conceptualization of a discovery pipeline involving ReLiEF fingerprints and TMAP visualization. So we start off here on the left with a list of potential receptors and potential ligands. Starting with the receptors, we can curate or generate structures using X-ray, cryo-EM, NMR, homology modeling, AlphaFold, for example, with mutations introduced. We can then clean and standardize those structures and enumerate their states, then apply short simulations and binding site prospecting to then generate ReLiEF fingerprints and TMAPs, associated TMAPs. And then we can really bias towards diversity maximization to generate a diverse structure set. With the ligands, we can clean and standardize those and enumerate their states. And then we do docking and scoring via this matrix of receptors and ligands. And associated with this matrix is going to be a TMAP. And we can color, for example, the TMAP based on score, with the idea being that we find these clusters here that have similar score, then those are probably regions we want to explore further and would represent structure-activity relationship perhaps. And so what's really exciting to point out here is that TMAP enables two things simultaneously. One, because of the fingerprints that are generated, those can be introduced into machine learning readily. And then because it's a visualization technique, med chemists, for example, could be looking at these clusters and structure-activity relationship at the same time, so in tandem with the machine learning. And I think that's something that's very exciting and a possibility to explore further. So there are a lot of ideas. I'm working on this. I continue to be working on it. So please stay tuned and please reach out to me if you have any questions. Thank you.